So what if I told you that the key to predicting a lung cancer patient's survival wasn't some fresh invasive tissue biopsy?
Right, which is what we've always relied on.
Exactly. But what if instead it was an AI examining a chest x-ray of a tumor that never actually existed?
It sounds completely ridiculous at first, I know.
It really does. Welcome back, Trailblazers, to the Digital Pathology Podcast. Today, we're doing a journal club-style deep dive into something that is, frankly, going to change how you practice medicine.
Yeah, we're talking about computational hallucinations or, well, synthetically generated medical data.
Which is ironic, right? Because as healthcare professionals, you spend your whole career trying to eliminate artifacts from your imaging.
Oh, absolutely. We hate anomalies. And now we're intentionally generating them to train our most advanced diagnostic tools.
It's wild, but as we'll get into, it's kind of the only mathematical way to solve the data scarcity problem when you're dealing with rare diseases.
It really is.
So, the source material for our deep dive today, and it's a dense one, but an absolute must-read, is a review article from the July 2026 issue of the journal Cancer Letters. It's volume 649.
A fantastic issue, by the way.
Oh, definitely. The piece is titled Artificial Intelligence in Clinical Oncology, Multimodal Integration and Translational Development.
And we really have to give credit to the author team here, because synthesizing this much data is, it's a massive effort.
It's huge. Let me make sure I get the whole team here. It's Ruichong Lin, Zhenhui Zhao, Zhonghai Liu, Jin Kang, Kang Zhang, Xiaoying Huang, and Yunfang Yu.
They basically mapped out the very bleeding edge of what's computationally and clinically possible right now in oncology.
And it comes not a moment too soon, right? The urgency they lay out at the start of the paper is just stark.
Yeah, the numbers are terrifying. They highlight that in 2022, we saw about 20 million new cancer cases globally.
Which is already a staggering burden on the system.
Right, but the projection is a 77% increase in new cases by the year 2050.
Wait, 77%?
Yes. A 77% increase means our current physical infrastructure, like our labs, our pathologists, the oncology wards, they will literally break under that volume.
You can't just build hospitals or train specialists fast enough to meet a curve like that.
Exactly, which is why AI in this context, it's not just some theoretical computer science experiment anymore.
It's a necessity.
It's a critical lifeline. For survival management, resource distribution, all of it, we are basically forced to fundamentally rethink how we extract diagnostic value from the data we already have.
So let's talk about the data we already have, because as a trailblazer listening to this, you know the immediate bottleneck you face every time you open a patient's chart.
Oh, the fragmentation.
Yes, the data fragmentation. In a modern oncology workup, you've got radiology doing the macro structure, right?
Right, the scans.
Then digital pathology is analyzing the microtissue, genomics is sequencing the molecular drivers, and then you have the longitudinal EHR tracking the patient's history.
But they all exist in absolute silos.
Completely isolated. It's like, I don't know, it's like trying to evaluate a patient's overall health by only looking at their cholesterol, while totally ignoring their blood pressure, their diet, their family history.
That's a great analogy. You get an answer, but you definitely don't get the whole truth.
Right, and traditional AI systems kind of have the same problem, don't they?
They do. Single-modality AI can only process one specific architecture of data at a time. So an AI trained strictly on MRI pixel gradients is entirely blind to the text-based clinical history.
Or the genetic sequencing.
Exactly. It evaluates the patient with profound tunnel vision.
So if a tumor doesn't exist in a vacuum because obviously it has a systemic biological impact, an AI that can't read the blood work alongside the tissue sample is just partially blind.
Which brings us to the core solution the paper proposes, which is multimodal integration.
Achieving cross-modal reasoning.
Right. And to really grasp how researchers are pulling this off, the authors give this fascinating clinical example regarding central nervous system tumors.
Oh yeah, CNS tumors like diffuse gliomas. They're notoriously ambiguous, right? Like trying to classify them using traditional histology alone is a nightmare.
It is. You almost always need complex molecular profiling to get an accurate subtype.
Which takes time.
Time the patient might not have. But the paper highlights this framework called NEP-SDA. It integrates spatial transcriptomics with graph neural networks, or GNNs.
Okay, let's unpack that, because we already use spatial transcriptomics to see exactly where specific gene expression is happening physically in the tissue slice.
Right, we know the where and the what.
But how does NEPSTA actually compute those two entirely different biological concepts together?
Well, it comes down to how a graph neural network mathematically perceives the world.
Yeah,
A standard image AI just looks at a flat grid of pixels.
like a photograph.
Exactly.
Yeah.
But a GNN analyzes networks. It turns the tumor microenvironment into a mathematical topology.
A topology, so it's looking at relationships.
Right. It treats individual cells as nodes, and then the physical proximity or the signaling pathways between those cells as edges.
Oh, wow. It's literally mapping the social network of the tumor.
That's a highly accurate way to frame it, actually.
Mhm.
So when you feed the special transcriptomics data into that GNN, the algorithm isn't just seeing that, hey, a specific oncogene is turned on.
It's seeing who that oncogene is talking to.
Exactly. It calculates how that oncogene's expression influences the neighboring immune cells based on their physical network connections.
That is mind-blowing.
It is. By synthesizing the geographic architecture of the tissue with its biological expression simultaneously, NEPSTA can molecularly subtype those difficult CNS tumors with incredible accuracy.
And it completely bypasses the traditional time-intensive molecular testing workflow,
Minutes instead of weeks.
which is incredible.
Yeah.
But it does bring up a major logistical hurdle for me. Fusing spatial data and cellular maps makes intuitive biological sense. They are both physical maps, essentially.
Sure.
But practically speaking, taking a 3D CT scan matrix and a text-based laboratory report, how does an algorithm mathematically reconcile those totally different data architectures?
Without just mashing them together into white noise.
Exactly. If you just stack a text file on top of an image file, doesn't the model just collapse under the weight of those conflicting dimensions?
It does, and that is the core computational challenge of multimodal AI. You can't just stack data. The breakthrough that Lin and colleagues highlight relies on something called cross-modal attention mechanisms.
Cross-modal attention. This is utilized in advanced transformer architectures, right?
Yes, heavily utilized.
Let's visualize that mechanism, because I think I have an analogy for this. It sounds a bit like an conductor managing a massive symphony orchestra.
Okay, I like where this is going.
The conductor doesn't just let the brass and the strings play over each other at maximum volume the whole time. If a specific cue happens in the sheet music, the conductor actively mutes the woodwinds and amplifies the strings.
That is the perfect mechanism-based analogy. The attention mechanism mathematically trains the AI to dynamically weigh the importance of different data types against each other in real time.
So it's actively shifting focus.
Exactly. The AI learns that if, say, demographic factor X and clinical symptom Y are present in the text data, it needs to direct its computational attention to a very specific pixel pattern in the imaging data.
And the paper points to a specific model called LUCD that does exactly this.
Yes, LUCD is a prime example of this seamless cross-modal attention. It integrates CT imaging, clinical symptoms, laboratory test results, and demographic info all at once.
So it essentially mimics the deductive, multi-layered reasoning of a multidisciplinary tumor board.
It really does. And the clinical application they outline for LUCD is just staggering. It can predict EGFR gene mutations and survival outcomes in lung cancer patients entirely non-invasively.
Wait, really? Non-invasively?
Completely. Think about a patient with a lung tumor that is positioned in a way that makes a physical biopsy highly dangerous or even impossible.
Which happens all the time in the clinic.
Exactly. LUCD fundamentally changes that diagnostic timeline. It looks at the CT scan, cross-references the routine labs, reads the clinical chart, and computes a highly probable molecular subtype without ever touching a scalpel.
It leverages the multimodal data the patient has already generated to extract insights we thought required physical tissue extraction. That's revolutionary for patient care.
It is. But to achieve that level of deductive reasoning, models like LUCD require an astronomical amount of meticulously annotated training data.
Right. And that exposes a massive vulnerability in how we currently build these systems.
The data bottleneck.
Yeah. It's relatively straightforward to gather, 1 million perfect lung scans for a highly prevalent disease if you are at a well-funded research institute.
Sure, the common cases are easy to find.
But as trailblazers, you are seeing rare presentations in the clinic. If the AI hasn't seen a rare tumor phenotype 1,000 times in its training data, its predictive accuracy just plummets, doesn't it?
It absolutely drops off a cliff. This is the data scarcity problem, and it's where the authors introduce one of the most radical and honestly controversial solutions in the review.
Which is using generative AI to synthesize the missing data.
Yes, they detail a foundation model called MINIM.
Okay, generating fake medical images to train clinical algorithms feels like walking on a tightrope to me.
It sounds incredibly risky.
Right. How does Minim translate a text prompt into a biologically accurate optical CT or fundus image without hallucinating fatal anatomical errors? Because if it makes a mistake, it corrupts the downstream AI.
Well, it avoids that by relying on highly constrained latent diffusion models. When MINIM generates a synthetic image, it isn't just, copy-pasting pixels from other scans it's seen.
It's not a collage.
Not at all. It has actually learned the underlying mathematical distribution of what biological tissue is, constrained by the strict laws of medical physics.
Okay, so it understands the rules of anatomy.
Precisely. So when you give it textual clinical parameters for a rare tumor, MINIM navigates that mathematical space to generate a statistically valid photorealistic scan of a pathology that fits all the physical rules.
Even if that specific patient doesn't actually exist.
Exactly.
So by feeding these synthetically generated images of rare cases into the training pipeline, we're basically forcing the diagnostic AI to learn the defining physiological features of the disease.
Right, rather than just overfitting its logic to the most common presentations it normally sees.
That makes a lot of sense, but does it actually work in practice?
It does, and the authors ground this in hard clinical results. They cite a lung cancer study where researchers utilized synthetic images generated by MINIM to supplement their limited real-world dataset.
And what happened?
Balancing a training set with the synthetic data didn't just improve the model's confidence scores in some theoretical vacuum. It directly improved the AI's accuracy in detecting real-world EGFR mutations, which subsequently refined their 5-year survival rate predictions. The computational hallucinations effectively bridge the gap in physical data.
Okay, so MINIM handles the synthesis of complex imaging, but oncology relies just as heavily on unstructured text, right? The notes hidden in clinical charts and pathology reports.
Oh, the text data is massive, and the review dedicates significant focus to the evolution of large language models, or LLMs, transitioning into this specific space.
Because general LLMs, the ones we all use on our phones, they're good, but they aren't doctors.
Right. The shift here is moving from general token prediction to specialized clinical reasoning.
Okay, explain the difference there.
Well, general LLMs are trained broadly on the entire internet. They are excellent at summarizing text, but they lack the constrained, rigorous logic required for medicine.
They guess what sounds right, not what is medically accurate.
Exactly. The authors contrast those with the new wave of medical-specific foundation models, like MedPollum and MedFound.
It's like the difference between asking a really well-read librarian for medical advice versus consulting a specialized attending physician.
I love that analogy.
The librarian knows where the words are, but the attending understands the clinical context. How does the underlying mechanism of MedPollum actually differ from a standard model when it reads an oncology chart?
So a general model looks at the chart and
Yeah.
basically predicts the most statistically likely next word to generate a readable summary. But MedPollum is structurally grounded in peer-reviewed clinical literature and medical ontologies.
It's built on a different foundation.
Entirely.
Right.
When it reads an EHR, it isn't just summarizing, it is actively cross-referencing the patient's nuanced history with current genomic databases.
So it's synthesizing new insights.
Right. It can extract an obscure oncogenic driver from a pathology report, match it against ongoing clinical trials, and then output a highly specific therapeutic plan with cited rationales.
It shifts the AI from being a passive document reader to an active participant in therapeutic strategy.
Exactly. It's a partner in the clinic.
Well, let's bring this back to the reality of the clinic then, because this all sounds amazing. We have LUCD performing non-invasive molecular subtyping using attention mechanisms. We have MANIM generating synthetic images to overcome data scarcity. We have MedPollum reasoning through complex patient charts.
It sounds like we solved everything, doesn't it?
It does. So if the technology is this good on paper, what is stopping a trailblazer listening to this right now from deploying it in their clinic tomorrow? Why aren't these multimodal systems integrated everywhere?
Yeah. Here is where we need a serious reality check. The paper is remarkably transparent about the translation gap between benchmark success and bedside deployment.
Lin and the team detail several critical roadblocks, right?
They do. And the most mathematically frustrating one is known as generalizability under distribution shift.
Distribution shift. Okay, AI models are notorious for looking flawless in retrospective lab studies and then failing completely when they hit the real world. Let's break down the mechanics of a distribution shift. What is actually causing the algorithm to stumble? Well, we have to remember that AI does not possess true medical comprehension, right? It recognizes complex statistical patterns.
Right. It's just really good at math.
Exactly. So imagine a multimodal model trained exclusively on data from a massive urban academic medical center. The AI learns to predict outcomes with 99% accuracy based on the specific pixel intensity gradients generated by that hospital's brand of high-end CT scanners.
Okay.
And it combines that with the baseline health metrics of an urban demographic.
So the AI has optimized itself perfectly for that specific environmental setup.
Exactly. Now deploy that exact same algorithm to a smaller, rural community clinic.
Different environment entirely.
Right. The community clinic uses a different manufacturer for their CT scanners.
Which apply a slightly different contrast calibration, maybe.
Yes. And the patient population has different baseline comorbidities. Mathematically, the underlying distribution of the data has shifted.
So the AI gets confused completely.
Hmm.
The AI, relying on the pixel gradients it memorized from the urban hospital, encounters this new scanner's output and confidently predicts the entirely wrong molecular subtype.
Because it learned a localized shortcut instead of a universal biological truth.
Yes, it memorized the camera, not the disease. And identifying these hidden stratifications before they harm a patient is incredibly difficult.
Which is why the authors emphasize the absolute necessity of prospective validation.
Yes. Retrospective benchmarking, where you feed an AI historical data and see if it predicts what we already know happened, is mathematically simple.
It's like predicting yesterday's weather.
Exactly. But deploying that model in a double-blind, randomized controlled trial where it informs real-time clinical decision-making across disparate healthcare networks, that is a massive logistical challenge.
And beyond the trials, it's a profound infrastructural bottleneck, too. I mean, running a 100 billion parameter multimodal transformer requires an astonishing amount of computational power.
Most standard hospital IT systems are simply not equipped to process continuous cross-modal attention mechanisms locally without intense latency issues.
You can't just run this on the laptop at the nurse's station.
No, scalability is a very real physical limit right now. We're asking local clinical servers to essentially perform supercomputing tasks.
And the cloud isn't a simple fix either, right?
Not with global data privacy regulations. The authors make it clear that until we can compress these models or build dedicated, highly secure medical inference networks, the deployment will remain limited to massive academic centers.
On top of that, there are the ethical and practical concerns of embedded biases in real-world data. If the AI is trained on biased data, it's going to automate those disparities.
Exactly. The overarching thesis of the paper becomes very clear. Pushing an AI's accuracy score from 95 to 98% on a static, curated dataset is no longer the primary goal.
It's vanity metrics at this point.
It really is. The entire field must transition away from performance-driven benchmarking and focus entirely on engineering clinically trustworthy, robust models that can survive the messy, uncalibrated reality of a real hospital environment.
Because trust is the metric that actually matters when a patient's treatment plan is on the line.
Absolutely.
So, synthesizing everything we've pulled from this extensive review, the fragmentation of oncology data, keeping scans, genomes, and charts in isolated silos, is mathematically limiting our ability to treat the whole patient.
Right. And we covered how cross-modal attention allows transformer models to dynamically fuse that data, unlocking non-invasive molecular insights.
Like predicting mutations without a biopsy. And we examine how synthetic generation via MINIM provides the missing mathematical links for rare diseases,
while confronting the very real engineering hurdles, like distribution shifts, that prevent immediate clinical deployment.
So as you trailblazers look toward your own practice, understanding these mechanisms isn't just about keeping up with technology. It is about preparing for a fundamental shift in multidisciplinary cancer management.
The shift is already happening.
It is. But before we wrap up this deep dive, there is one final staggering concept briefly introduced at the conclusion of the paper that rethinks our entire approach to disease modeling.
Oh, this is the part that really blew my mind. It's a paradigm shift in how we view the timeline of therapeutic intervention.
The authors point toward the impending reality of the virtual cell and mechanistic foundation frameworks.
Right.
Consider this as you review your next patient chart. What if we no longer test a novel targeted therapy's efficacy by running a years-long human trial, or even by analyzing a physical tissue biopsy in a lab?
It sounds like science fiction.
It does. But if multimodal AI can perfectly map the spatial transcriptomics, the real-time imaging, and the complete clinical history, it theoretically possesses all the parameters necessary to build a perfectly accurate, computationally interactive digital twin of a patient's specific cancer.
A virtual cell simulation.
Right. You could simulate the complex molecular interaction between a new drug and the tumor environment entirely in silico.
Without ever touching the patient.
Exactly. You could test thousands of different dosing regimens on the digital twin in a single afternoon, observing mechanistic cellular responses mathematically before ever administering a drop of medicine to the physical patient.
It takes the cross-modal reasoning we discussed today and applies it to the dimension of time and simulation.
It completely alters the speed and safety of translational research and personalized medicine.
It moves us from a fragmented view of disease to a fully simulated, navigable digital reality.
It is a massive concept to process, and it challenges everything about how we currently design treatment protocols. So, something for you to mull over today. Thank you for joining us for this deep dive into the underlying architecture of clinical oncology's future.
Keep pushing those boundaries.
Keep questioning the mechanisms behind your data, and we will catch you on the next deep dive.